Welcome back to this next video in which we are discussing inflammatory bowel disease. Okay, so we're currently in the process of studying the pathophysiology of inflammatory bowel disease, and the point in the story that we've got to so far is that our individual who is going to suffer from inflammatory bowel disease is going to develop a clone of CD4 positive T cells that is directed against a peptide fragment of one of the proteins of a intestinal commensal. And we discussed in the previous video that we don't really understand why individuals who are going to develop inflammatory bowel disease do develop these CD4 positive T cell clones that are directed against commensals, and why individuals who aren't going to develop inflammatory bowel disease don't develop those CD4 positive T cell clones directed against commensals. We don't understand that at present. What we do know is that both genetic factors and environmental and developmental factors play a role in determining what CD4 positive T cell clones you actually end up with. But we don't really understand what those genetic factors and those environmental factors are yet, and how they actually determine which CD4 positive T cell clones you end up with. So I can't really give you any more insight into that. But we do know that it's not merely just genetic factors. Environmental factors do play a role in determining which CD4 positive T cell clones you end up with as well. Now, an important point that I didn't tell you about in the previous video, but which I need to tell you about now, is the fact that actually the CD4 positive T cell clones that your body has present within it is determined during childhood. And once you hit adolescence and then adulthood, you don't change your population of CD4 positive T cell clones. You don't get any new CD4 positive T cell clones. The clones that you have are fixed after that point. And the reason is that to create brand new CD4 positive T cell clones, you require a gland called the thymus gland, and I'll just write its name up here. So, in children, there is this gland called the thymus gland that is present in the mediastinum just behind the sternum. However, once you hit adolescence and then go into the adulthood years, that gland has atrophied away and been replaced by fat tissue. So the thymus gland is a gland that's only present in childhood, and this gland is absolutely essential for creating brand new CD4 positive T cells and also CD8 positive T cells, but we're not interested in those ones. It's absolutely essential for creating brand new CD4 positive T cells of a new type, i.e. that are directed against brand new peptide fragments. So, once you hit adolescence and adulthood, this thymus gland has gone, and actually you don't create any new CD4 positive T cell clones. So, the question then arises, how do you maintain your population of CD4 positive T cells? If the thymus gland has gone and you're not creating any brand new ones, how do you maintain your population? Do they just live for the rest of your life? The answer is no, they don't quite live that long. CD4 positive T cells do have a long lifetime, however they don't generally last your entire lifetime, so they will eventually die. So then how do we maintain our population? The answer is that they divide at an extremely slow rate. So the CD4 positive T cells that you have in your body, they'll be dividing at an extremely slow rate. And this extremely slow rate of division is how they maintain their population as CD4 positive T cells are dying. And the idea then is that you maintain all of the same clones that you developed during childhood by this gradual division to replace the ones that are dying, but you don't create any new clones. So overall, what have I told you there? Once we're in adolescence and adulthood, we don't create new clones of CD4 positive T cells. Our immune system is built at that point as far as CD4 positive T cells are concerned, and we just maintain it after that. So, what that means is that this individual who is going to develop inflammatory bowel disease actually developed the CD4 positive T cell clone that is capable of attacking one of the commensals when they were a child. So, a question then arises. they have this CD4 positive T cell clone from when they were a child, but they don't develop the disease until their second or third decade of life. So they might not develop the disease until they're 30 years old, maybe. So why is the delay? Why can you have this CD4 positive T cell clone directed against a peptide fragment of a protein of one of the commensals, and yet you don't develop the disease till years later? Well, again, we only have a theory as to why this is, so it's just speculation. But there is a lot of interest into the concept that the microbiota evolves. So I'll just write this down here. So the answer to this is speculated to be evolution of the microbiota of the gut. So I might put of gut microbiota. So firstly, what do I mean by microbiota? Microbiota is a nice word that I'll use again throughout this video, so it's a good word to introduce. This means the collection of commensals within your intestine. So the gut microbiota is the collection of all the commensals, all the life that you have within the lumen of your intestine. So it just refers to all the commensals. Evolution means change over time, so evolution of gut microbiota means that the gut microbiota, the collection of commensals that you have within your intestinal lumen, is going to change over time. So this is believed to be the answer as to why you don't develop the disease potentially till years after you've actually had the CD4 positive T cell clone available. Because the gut microbiota is not fixed throughout your lifetime. The collection of commensals that you have will change as you get older, and there's a huge amount of interest into studying this at present. So it is speculated then that if you're not going to develop the disease until years after developing the CD4 positive T cell clone, the reason for the delay is that at the time when the CD4 positive T cell clone develops, the commensal that it's targeted against is not present in any sort of significant level within the intestine. And then as you get older, the microbiota evolves, and that commensal that you can attack becomes much more prevalent. So it's got a much higher population within the intestine, and that is then when you will actually start initiating this immune response that is laid dormant for many years. Okay, so to summarise what I've said then, we have our individual who in childhood is going to develop this CD4 positive T cell clone that can attack a commensal. The reason then there is that delay between developing the T cell clone and actually developing inflammatory bowel disease is believed to be that the commensal might not actually be present in the intestinal lumen in any high amount during childhood, and then might become more prevalent as you go into adulthood. And this is the concept of evolution of the gut microbiota. And then once it's present in a high amount during the within the intestine, that's when you'll actually develop inflammatory bowel disease. Okay, so we have more to say though. We want to now understand how we're actually going to end up activating these CD4 positive T cell clones that we've got. So let's say we now have the commensal that we are capable of attacking present in a high amount within the gut. We want to understand how do you actually then activate the CD4 positive T cell clone that you developed during childhood. So let's study the story in a little bit more detail. Okay, so this requires us to study something called immunosurveillance that is occurring within the intestine, and this requires me to tell you a little bit more about the cells of the epithelium of the intestine. So we're going to discuss something called immunosurveillance. So of course, surveillance means watching. Immuno means to do with the immune system. So this is going to be the immune system watching the intestine. And this is all going to be about trying to spot when there are pathogens present within the intestine. So we've studied these commensals. We've talked about how within the intestinal lumen there are loads of microorganisms that are actually not harmful and actually help us with digestion. However, that isn't always the case. Sometimes you can get microorganisms within the intestine that are dangerous, and if they weren't dealt with, would start invading the intestinal tissue and cause huge damage to our body. Therefore, within the intestine, we need to be continually looking out for signs of pathogens. And this is the concept of immunosurveillance. And the way I'm going to talk you through this is I'm going to talk you through how this would work in terms of pathogens, i.e. its physiological function, its good function, the reason we have this. And then I'm going to show you what's going to happen in terms of the commensals, both in a person who's not going to develop inflammatory bowel disease, and then we'll see what would happen, what would go wrong in an individual who does have inflammatory bowel disease, and how this is going to lead to the inflammation. And remember, in the case of a pathogen, the innate immune response comes first and then the adaptive immune response follows. In the case of inflammatory bowel disease, it's going to be the innate immune response doesn't attack. It knows that this isn't something that should be attacked. Whereas the adaptive immune response does attack, and it overpowers the innate immune response and tells the innate immune response you're going to attack whether you like it or not. Okay, so let's understand this then. So, back up to my picture of the intestinal epithelium here. So, we can be talking now about the intestinal epithelium of the large intestine or the small intestine. So I've told you about how there are the major epithelial cells, which are the enterocytes, and I'm sorry the word has disappeared, but it should be there, the enterocytes within the small intestine and the colonic epithelial cells within the large intestine. And I've also told you about the goblet cells, and I've told you about the intestinal stem cells. I'm now going to tell you about another type of epithelial cell that is one of the more niche epithelial cells that I didn't mention initially, but which is another one that can be produced by differentiating cells from the intestinal stem cells. And these are called M cells, and these are extremely important in immunosurveillance within the intestine, which is why we are going to study them. So, allow me just to draw a picture then. So, I'm going to draw a little piece of the epithelium here. So, this is representing a piece of basement membrane, of course, in red there. And then we'll have some epithelial cells on the basement membrane. So I'll draw these here. So this can just be a normal epithelial cell, either an enterocyte if we're talking about the small intestine, or a colonic epithelial cell if we're talking about the large intestine. And here's another one. And then I'm going to have one of these new types of cells in the middle there. And I think I'll colour it in some fancy colour. So here in blue, still a cuboidal cell to fit in with the rest. This is one of these cells that is going to be part of the immunosurveillance, and which is called an M cell. Okay, so first piece of information about immunosurveillance. I'm adding in another type of epithelial cell to my intestinal epithelium, and this is called these M cells, and these M cells are going to be part of the immunosurveillance. Now you might think, well, maybe the M cells are actually going to look out for pathogens themselves. Maybe they are the sentinel cells. So this is a very important concept from immunology, so I'll write it down. So in immunology, we talk about a collection of cells which are called sentinel cells. And sentinel means guard. So these are cells that are on guard. These are cells that are present actually in all the tissues within your body and are continually looking out for signs of pathogens. And if they find signs of pathogens, they set off the inflammatory response. They call in the innate immune response. So I'm talking about these M cells, and you might think, well, maybe these M cells are a sentinel cell. The M cells are not sentinel cells. The sentinel cells in the gastrointestinal tract are the same sort of cells that we have everywhere else in the body, namely resident macrophages and dendritic cells, and they are in the lamina propria underneath the epithelium. So I'll draw these in here. So firstly, this great big blob of a cell, this is a macrophage. Now, macrophages are a major cell that you bring in as part of the inflammatory response. However, some of them are permanently positioned in tissues and function as sentinel cells, so we call these resident macrophages. By the way, a useful abbreviation for macrophage is to abbreviate it down to M and then phi. So I'll use that from now on when I'm referring to macrophages, just to avoid having to write out macrophage. So, all over your body, you have resident macrophages that are performing a sentinel role. They stand on guard, and if they find signs of a pathogen, they set off the inflammatory response. So in the intestine, both the small intestine and the large intestine, you're going to have, in the lamina propria layer, resident macrophages. The other major type of sentinel cell that, again, you find all over the body are dendritic cells. And these are, again, a classical cell, and they have these dendrite processes. So you can see that I've drawn it in this sort of star shape. They have the dendrites coming off the major cell body here. And this is to make the dendritic cell have a large surface area so that it can extend these dendrites into loads of far-off portions of tissue to look for pathogens in those far-off portions of tissues. So this here, this is a dendritic cell. And for short, I'll abbreviate dendritic cell down to DC. Okay, so again, resident macrophages and dendritic cells, these are cells that we call sentinel cells. You find them all over the body. They are standing guard looking for pathogens. And in the intestine, they are present within the lamina propria. Now, the problem is the pathogens are most likely going to be within the lumen of the intestine rather than within the lamina propria. Of course, if they are in the lamina propria, that's a major problem and the sentinel cells will catch them. However, a lot of the pathogens will start off in the lumen of the intestine and then will go into the intestinal wall. So it would be good if we could catch them whilst they were still in the lumen rather than waiting for them to actually invade into the intestinal wall. And this is where the M cells come in. The M cells take samples of microorganisms that are in the lumen of the intestine and hand them into the lamina propria where there are sentinel cells, and the sentinel cells will then phagocytose these pathogens, have a good, well, microorganisms I should say, because they're not necessarily pathogens. We're going to do this for commensals as well as pathogens. Have a good look at them and decide whether they are pathogens. And if they decide that they are pathogens, they will initiate the inflammatory response. So let me just explain this in a little bit more detail. So, in red over here, this little blob can represent some microorganism. And notice I'm saying microorganism at this point rather than pathogen, because as I say, we're going to do this not just for pathogens, but also for the commensals. So this is a microorganism. So the M cells that are in the intestinal epithelium, they can endocytose little samples of microorganisms into little vesicles, move them across their cytoplasm, and then fuse that vesicle with their basolateral membrane to release the pathogen into the lamina propria where the sentinel cells are present. So let me just draw an M cell a little bit bigger here. So this is my M cell taken out and drawn much larger. And I'll put its nucleus here so it's nicely out of the way. So, if we have our microorganism, again up here in red, what can happen is the M cell can endocytose that microorganism into a little vesicle, like so. So here is its vesicle, and then the microorganism will be in that little vesicle, and then that little vesicle can move through the cytoplasm to the other side of the M cell, fuse with the membrane on the other side, and release the little microorganism into the lamina propria, right onto where there will be some resident macrophages and dendritic cells waiting for that microorganism. And what's now going to happen is these are going to phagocytose that microorganism and break it down. So they will endocytose it into a vesicle and break it down. And whilst they're doing that, they will be having a good look at that microorganism to see if there are signs that that microorganism is dangerous. So what do I mean by this? So the sentinel cells, the resident macrophages and the dendritic cells, they have a huge number of receptors that can look for certain molecules that are associated with pathogens. Now, there are absolutely loads of molecules that can be associated with pathogens, and for short, we have an umbrella term that we use for all of them, which is pathogen-associated molecular patterns, which for short we abbreviate down to PAMPs. So pathogen-associated molecular patterns, which, as I say, for short, we abbreviate down to PAMPs. So, pathogens have a huge number of molecules that no human cell would ever produce, and which our sentinel cells have learnt are associated with pathogens. There are absolutely loads of these, and we create this umbrella term, pathogen-associated molecular patterns, or PAMPs, which covers all of these molecules that are associated with pathogens. And the sentinel cells, the resident macrophages and the dendritic cells, they have receptors that can recognize different pathogen-associated molecular patterns. And the different pathogen-associated molecular patterns will, of course, have different receptors. But again, we have an umbrella term for these receptors that recognize pathogen-associated molecular patterns, and we call them pattern recognition receptors, which for short we can abbreviate down to PRRs. So let me talk you through that again. So, pathogens, bad microorganisms that could cause damage to our bodies. There are loads of molecules that are associated with pathogens and which our sentinel cells know are associated with pathogens. These collectively we call pathogen-associated molecular patterns, or PAMPs. Now, the sentinel cells, the way they can recognize pathogen-associated molecular patterns is by having receptors for pathogen-associated molecular patterns. And of course, the different pathogen-associated molecular patterns will need different receptors, but again, collectively, we refer to these as the pattern recognition receptors. We have an umbrella term, which is the PRRs. So, when these sentinel cells here are handed this microorganism, they will phagocytose it and they will be looking at it very carefully. Their pattern recognition receptors will be looking for any signs of pathogen-associated molecular patterns on that microorganism. And if they find any pathogen-associated molecular patterns, they will know that that microorganism is a pathogen, and they will then initiate the inflammatory response. Okay, and the inflammatory response is, of course, the innate immune response. So this is what would happen when we're actually dealing with a proper pathogen. If this microorganism was a pathogen, then the sentinel cells would recognize it as a pathogen by recognizing its pathogen-associated molecular patterns using their pattern recognition receptors, and they would initiate the innate immune response, or the inflammatory response. And the way in which they initiate the inflammatory response is by releasing a bunch of very powerful molecules, pro-inflammatory molecules. And for now, the two major pro-inflammatory molecules that they can release are interleukin-1, abbreviated down to IL-1, and tumor necrosis factor alpha, TNF-alpha. If you are going to learn just two pro-inflammatory mediators, these are the two to learn. So let me write out their names in full. So IL-1 in full, that is interleukin-1. Inter meaning between, leukin referring to white blood cells, so it's a molecule that goes in between cells of the immune system. And TNF-alpha, that's short for tumor necrosis factor alpha. TNF-alpha. So these, as I say, are major examples of pro-inflammatory molecules. There are loads and loads more pro-inflammatory molecules, but these are the two to learn above all the others. Okay, right. And now both of these are proteins. And you might see another word used for these. You might see the word cytokine used. Now, a cytokine is a name for a molecule that is released by a cell of the immune system, which is a protein, and which goes and changes the behavior of another cell. So, interleukin-1 and TNF-alpha, these are released by cells of the immune system. These sentinel cells are cells of the immune system. They are both proteins, and they are going to go and change the behavior of other cells. So you can describe these as cytokines. However, there are other pro-inflammatory mediators that are released, less important than these two, which are not proteins, and therefore you would not call them pro-inflammatory cytokines. So it's broader to use the word or the term pro-inflammatory mediators than pro-inflammatory cytokines, because cytokines only really covers the protein mediators. Okay, so what are these molecules going to do then? Well, they're going to trigger inflammation. What is inflammation? Well, we discussed this right at the start of the video. Inflammation is fundamentally a response of the microvasculature. It's a response of the tiny little blood vessels, the arterioles, the capillaries, and the venules. And this response in those microvasculature blood vessels is going to allow us to recruit troops from the blood. Remember, we think that this microorganism is a pathogen. At the moment, we're not talking about the pathophysiology of inflammatory bowel disease. I'm telling you about the normal response to pathogens, and then we'll contrast that to what's going to happen in inflammatory bowel disease. So at the moment, we are assuming that this truly is a pathogen, and it's been passed to the sentinel cells. The sentinel cells have recognized it as a pathogen, and they have released these pro-inflammatory mediators that are going to cause inflammation. So we want to bring in troops from the bloodstream that is going to help us deal with this pathogen, clear this pathogen. So we're going to fundamentally have a response that is mediated by the microvasculature. So let me put this here. So the pro-inflammatory mediators are going to work on the microvasculature, and the microvasculature will start recruiting troops from the bloodstream. And of course, we know that the microvasculature is very close by. If I go back up here to my picture of the intestinal wall structure, here are here's an arteriole, here's a venule, loads of little capillaries in between the two. So these are the blood vessels that are going to now respond to the inflammatory mediators. And look, these are in the lamina propria, which is exactly where those sentinel cells are present and are going to be releasing these pro-inflammatory mediators. So the pro-inflammatory mediators don't have to diffuse far, they diffuse to the microvasculature, and they're going to cause an exudate to be formed, which, remember, is lots of fluid containing proteins from the bloodstream. And these proteins that come in from the bloodstream, they are going to be able to attack pathogens. Major examples would be the complement proteins. And you're also going to bring in white blood cells, which I'll abbreviate down to WBCs. And the major two types of white blood cells that you bring in as part of the innate immune response, or the inflammatory response, are neutrophils and macrophages. And both of these types of white blood cells are phagocytes. They can endocytose pathogens and then break them down and destroy them. Macrophages, we've already got a picture of a macrophage. They look exactly like these resident macrophages here. They're great big blobs of cells. Let me just draw you a picture of a neutrophil, because neutrophils look quite different. They're not as blobby as macrophages. They're more a round cell. And their nucleus is a funny shape. Their nucleus is multi-lobed, like so. So they don't have multiple nuclei, they have one nucleus, but their nucleus has this funny shape where it's split into separate lobes, and usually you have 3 separate lobes, as I've shown on this picture here. Okay, so this then is the inflammatory response. So I should just put here that this is inflammation, or the innate immune response. And as you can see, neutrophils and macrophages, the two major types of white blood cells that you bring in, and the proteins that you bring in from the blood, they are not specific at all to the pathogen. So this truly is the innate immune response. It's not specific at all. These cells can phagocytose any old pathogen, and the proteins will attack any old pathogen. It's not a unique response to that pathogen at all. Okay, so let me just summarize what I've told you, and then we'll return back to discussing what's going to happen in inflammatory bowel disease. Okay, so we have been talking about immunosurveillance that occurs within the intestine. So this is all about looking out for the presence of pathogens within the lumen of the intestine. So the way this works is that we have these M cells within the epithelium, and these M cells pass samples of microorganisms from the lumen of the intestine, down into the lamina propria where we have sentinel cells, namely resident macrophages and dendritic cells, which can have a good look at them and break those microorganisms down. And if they decide that that microorganism is a pathogen, they will release the pro-inflammatory mediators interleukin-1 and tumor necrosis factor alpha, and you will get inflammation occurring within the lamina propria, and these cells and the exudate will go also into the lumen and will be able to attack the pathogen as well. Okay, so you'll get inflammation occurring when there is a pathogen present. Now, of course, the M cells will do this for the commensals as well. Talking now about the commensals, so we'll have a commensal here now. The M cell will sample the commensals, pass them down into the lamina propria to the sentinel cells, the resident macrophages and the dendritic cells. And again, these sentinel cells will have a good look at that microorganism, that commensal microorganism this time, and they will decide that that is not a pathogen, because it doesn't have the pathogen-associated molecular patterns. It's a commensal. Remember, the innate immune response, even in inflammatory bowel disease, does not attack the commensals. It's the adaptive immune response that goes wrong. So these will not decide that that is a pathogen. They'll decide that it's perfectly all right. They'll still break it down, but they won't launch an inflammatory response, so you won't get interleukin-1 and tumor necrosis factor alpha released, and therefore you won't get inflammation occurring. And this is still what happens in inflammatory bowel disease. You don't get the innate immune response first, as you would do normally. Instead, you're going to get the adaptive immune response occurring. So let's now talk about how we're going to end up initiating the adaptive immune response. So the story of immunosurveillance does not end there. This is how we can initiate the innate immune response using the sentinel cells. But even when they don't initiate the innate immune response, these sentinel cells can go for a second opinion. I've told you that they're still going to break down the commensals that they've been handed as a sample by the M cells. And when they break them down, they will break down all the proteins of that commensal. They'll break the proteins down into little peptide fragments, and they're now going to take these peptide fragments to the adaptive immune response cells, to T cells, and they're going to just double check that none of these peptide fragments are from a pathogen that is dangerous. So they have examined the pathogen using their primitive pattern recognition receptors to try and see whether this microorganism is dangerous, and I apologize, I just called it a pathogen. They have looked at the microorganism using their primitive pattern recognition receptors to try and determine whether it's dangerous, and they've decided it's not dangerous. However, their pattern recognition receptors are not brilliant. They only have a limited number. Therefore, they only have a limited capacity to recognize pathogenic microorganisms. So they go for a second opinion just to make absolutely sure they're going to okay it with the cells of the adaptive immune response. So they will chop up all the proteins of the microorganism that they phagocytosed, and they'll present them on their surface to T cells. And, of course, if the microorganism is not harmful, if it's a commensal, then in the ideal world, no T cells should be directed against peptide fragments of the proteins of that microorganism, and therefore you should get no adaptive immune response. However, in inflammatory bowel disease, that's going to go wrong, and this double checking, this second opinion bit, is actually going to end up activating the adaptive immune response. So let me just go over this in a little bit more detail. So, firstly, we need to talk about how they're going to present the peptide fragments of the microorganism that they've chopped up. So, I've said that once they've phagocytosed the microorganism, they chop up all its proteins into little peptide fragments and they present them on their surface. Now, they have two options for how they can present the peptide fragments on their surface. They can present them on something called MHC class 1, or they can present them on something called MHC class 2. So let me just talk you through this. So there are these two ways in which these sentinel cells can present the peptide fragments to the T cells. And by the way, I should introduce you to another little piece of terminology. The sentinel cells, the resident macrophages and the dendritic cells are also what we call antigen-presenting cells. When we're talking about them looking out for pathogens using their primitive pattern recognition receptors and then initiating the innate immune response in response to activation of the pattern recognition receptors, we call them sentinel cells. When they now go to double check it with the adaptive immune response, we call them antigen presenting cells. So the A is for antigen, the P is for presenting, and then the C is for cells, because they are presenting fragments of potential antigens to the cells of the adaptive immune response. Okay, so back to MHC class 1 and MHC class 2. So firstly, what does MHC stand for? So it's a big long name, hence why we abbreviate it down normally to MHC. So MHC stands for Major Histocompatibility, and I think that's how you spell that. Complex, that's the C. And then you have major histocompatibility complex class 1 and major histocompatibility complex class 2. So our antigen-presenting cells, the resident macrophages and the dendritic cells, they are covered on their surface with MHC class 1 and MHC class 2 molecules. And this is my basic picture of it. It's completely out of proportion as far as the size of the cell is concerned, but it gets the point across. So it's a protein on the surface of the cell. Both of them are proteins on the surface of the cell. And look, you have here a little groove where you can put a peptide fragment, and this is how they can present peptide fragments. So when these antigen-presenting cells have chopped up the microorganism and they've chopped up all its proteins into little peptide fragments, how are they actually going to decide whether to put the peptide fragments on MHC class 1 or MHC class 2? So this is a really important thing to understand. You put peptide fragments of intracellular pathogens on MHC class 1, and you then go and present those peptide fragments to CD8 positive T cells, which, if you remember, are the T cells that mediate the intracellular adaptive immune response. Whereas, if you're dealing with an extracellular pathogen, you put the peptide fragments on MHC class 2 molecules, and you then present them to the CD4 positive T cells, which are the T cells of the extracellular adaptive immune response. So MHC class 1 goes with CD8 positive T cells and is part of the intracellular adaptive immune response. MHC class 2 goes with CD4 positive T cells and is part of the extracellular adaptive immune response. So of course MHC class 2 is the one that I'm interested in. So, how do the antigen-presenting cells know whether they have an intracellular or an extracellular microorganism here? Well, if they have phagocytosed the organism from the extracellular fluid, they know then that it's an extracellular organism, and then when they chop up its proteins into little fragments, they'll put those fragments on MHC class 2. So these got the pathogen from the extracellular fluid, so they know therefore it's extracellular. If instead they have phagocytosed a dying human cell that is full of an intracellular pathogen, and they have then chopped up the proteins of that intracellular pathogen, they will then put those peptide fragments on MHC class 1 because they know that those were intracellular pathogens because they phagocytosed a dying human cell that contains those pathogens. So that's how they can know the difference between an intracellular pathogen and an extracellular pathogen. It's all about how they actually found that pathogen. Did they find it in the extracellular fluid, or did they find it inside a dying human cell? Okay, so we are being handed extracellular microorganisms here by the M cells. Therefore, when we chop up the proteins of that extracellular microorganism, we will put its fragments on MHC class 2 protein complexes. Now, again, I'll repeat this. You do this for all microorganisms, whether it's a pathogen or whether it's a commensal. This is a second check, okay? Even if the sentinel cells have decided that it's not a pathogen, they're going to still go and cut up its proteins and put them on MHC class 2 and just check it with the adaptive immune response. And in people who don't develop inflammatory bowel disease, this is absolutely fine. This doesn't cause a problem at all because the adaptive immune response will agree with the innate immune response, it will agree with the sentinel cells, and it will say this is not a pathogen, do nothing. However, in inflammatory bowel disease, it's going to go wrong because you are going to have those CD4 positive T cells directed against peptide fragments of the proteins of your commensal, and therefore you are going to end up initiating an adaptive immune response that will then tell the innate immune response to attack as well. Okay, so we will phagocytose our microorganism, will chop up all its proteins, and will put the peptide fragments on the MHC class II molecules. What happens now? Well, once the sentinel cells have got loads of peptide fragments on the MHC class II molecules, they're going to go and present that to T cells. Now, I need to just stress a point here. This microorganism that we have phagocytosed, it will have a huge number of different proteins, and those proteins will have a huge number of different peptide fragments, and you can chop up those different proteins in loads of different ways. So that means that this is going to be covered in a huge number of different peptide fragments now, and it's now going to go and present all of those different peptide fragments to T cells and try and find a T cell that is going to actually be directed against one of those peptide fragments. So let me now show this. So, how is it going to actually find T cells? Well, T cells are actually present within the lamina propria of the intestinal wall. This is another part of the histology that I didn't go over when we went into our basic histology of the intestine up here. In the lamina propria, this portion of connective tissue underneath the basement membrane, you actually have lymphoid aggregates that are very similar to lymph nodes, effectively, within the wall of the intestine. And these are aggregates of T cells and also B cells as well. And you'll have T cells of both the different types, so CD4 positive T cells and CD8 positive T cells. So I don't know how I'm going to draw this. I will draw it in on the picture down here rather than in that picture up there. So, in the lamina propria then, this great big blob here, this is going to represent a lymphoid aggregate, and you have these dotted along the intestine. So if you take a section of intestine at the exact right place, you will potentially be able to see one of these lymphoid aggregates. And it's just an aggregate of loads and loads of B cells, loads and loads of T cells as well. So this is going to be a lymphoid aggregate. Okay, now there are special names for these lymphoid aggregates when you're in the ileum. So in the ileum, the lymphoid aggregates are called payers patches, and you might have heard of that before when you studied histology. So they're just lymphoid aggregates, loads and loads of T lymphocytes and B lymphocytes. And these are another place where lymphocytes can hang out. So remember I said that lymphocytes are not static. They can live in lymph nodes, they can live in the spleen, they can now live in these gut lymphoid aggregates, and I might actually give them their full name. These are gut lymphoid aggregates, and you don't just have them in the ileum. They're given a special name in the ileum. They're called Peyer's patches, and they're dotted along the length of the intestine, but you have them along the entire length of the intestine. All of the small intestine will have gut lymphoid aggregates, and in the large intestine as well, in the lamina propria, you'll have gut lymphoid aggregates. So lymphocytes can hang around in all of these places, and of course they can be in the blood as well. And lymphocytes do not stay put. It's not like one CD4 positive T cell will settle down in this gut lymphoid aggregate and stay there for the rest of its life. It can leave that lymphoid aggregate, go into the lymphatic vessels, drain back into the systemic circulation, and then it can go to some other lymphoid tissue. It can go to a lymph node, maybe up in the neck, or it can go to the spleen for a while. So lymphocytes are not static, they move around the body, and this means that we're continually rejigging the positions of our CD4 positive T cell clones. And of course, if you know something about immunology, you'll know that CD8 positive T cells have loads of different clones, and B cells have loads of different clones as well. So this means that we're continually rejigging our clones around, moving them around different parts of the body, so that all the different parts of the body will have the different clones present there. And that's important so that we can use these immune responses all over the body. I mean, we just wouldn't want one clone just being present in a certain location in the body for the rest of your life, and then not being usable if you get an infection somewhere else. So you do need to continually be mixing up your lymphocyte clones. Okay, right, so in the lamina propria then we have these gut lymphoid aggregates, and this is where these antigen-presenting cells are now going to go, and they're going to try and find CD4 positive T cells now to activate. So this can represent a CD4 positive T cell. So I'll remind you that they are presenting peptide fragments on the MHC class 2 molecules, and therefore they cannot activate the CD8 positive T cells. They require them presented on MHC class 1. So if you're presenting on MHC class 2, you can only activate CD4 positive T cells. So our antigen-presenting cell here, covered in peptide fragments of our microorganism, is now going to go to the lymphoid aggregates, and it's going to look for CD4 positive T cells that are directed against the peptide fragments that it is presenting, and if it finds them, then it will activate them, and therefore it will end up activating the adaptive immune response. Now, let's talk about what should happen then. So if this microorganism is a pathogen, then what will happen is the pathogen will be moved into the lamina propria by the M cell. It will then be phagocytosed by our sentinel cells here. The sentinel cells will recognize it as a pathogen. They will start the inflammatory response up by releasing interleukin-1 and TNF-alpha. They'll then go down to the lymphoid aggregates with all the different peptide fragments of the proteins of that pathogen on their surface, and they will activate CD4 positive T cells that are directed against that pathogen. Okay, so you get the innate immune response occurring first, and then you get the adaptive immune response occurring second, or being initiated second. We haven't gone through the adaptive immune response in full yet. Don't worry, we will complete that story up. If we're talking about a commensal and we're talking about an individual who's not going to develop inflammatory bowel disease, then what will happen is the commensal will be sampled by the M cells, it'll be looked at by the sentinel cells, they'll say, nope, this isn't a pathogen, no inflammatory response will occur. They'll then chop up the commensal's proteins, put its peptide fragments on their surface, bound to MHC class 2, and they'll go to the lymphoid aggregates, and they will find no CD4 positive T cells that are directed against any of those peptide fragments, and therefore you'll get no adaptive immune response. Fantastic, you do not get inflammatory bowel disease. However, in our individual who's going to develop inflammatory bowel disease, the commensal will come in from the M cell. The sentinel cells will still say, nope, this isn't a pathogen, no inflammatory response will occur, but now when they chop up all its proteins and put the fragments on MHC class 2 molecules and take this down to the lymphoid aggregates to present to CD4 positive T cells, they might just find a CD4 positive T cell that is directed against one of those peptide fragments, if indeed you have developed a CD4 positive T cell clone directed against a peptide fragment of a protein of this commensal. Now, of course, you might say, but think about this. This commensal, when it's been chopped up, when all of its proteins have been chopped up, this sentinel cell, this antigen-presenting cell, might end up chopping those proteins up in the wrong way and then might not get the correct peptide fragment, and therefore maybe we won't initiate the adaptive immune response because we're presenting the wrong peptide fragments simply because our antigen-presenting cell chopped them up in the wrong way. And I would agree with you. However, you've got to remember that this will be occurring continuously. You'll be getting more and more of these commensals moved in. This sampling process occurs continuously. Immunosurveillance doesn't just do the microorganism once and then say, let's never sample that microorganism again. It'll go on and on and on, and therefore eventually one of the antigen-presenting cells will end up chopping up the proteins in the correct way to get the peptide fragment that you actually have developed a CD4 positive T cell clone directed against, and then it might come down to this lymphoid aggregate and actually find that CD4 positive T cell. And again, another argument you might have is, well, what if the CD4 positive T cells directed against that peptide fragment aren't currently in that lymphoid aggregate? Maybe they're up in lymph nodes in the neck, so it might not actually find the correct CD4 positive T cell. Again, I would say absolutely, but we know that the lymphocyte clones shift around, so eventually what will happen is you will end up with one of the CD4 positive T cells directed against that peptide fragment in the correct lymphoid aggregate, and the sentinel cell has chopped up the proteins in the correct way so that it is presenting that peptide fragment, and therefore it will go down here and activate that CD4 positive T cell. Okay, we will halt the story there. We'll leave on a cliffhanger, and in the next video we'll see what now happens once you have activated the CD4 positive T cell directed against the peptide fragment of one of the proteins of the commensal.